Good afternoon, my dear students. My channel hasn't had any English videos for a long time. Today I am inspired to tell you about the main tests from Krok 1 base on the topic laboratory diagnosis of cholera and bacillary dysentery. Unfortunately, in Ukraine, due to the war, these diseases often began to appear in the occupied territories. The causative agents of cholera and bacillary dysentery have many common features, namely, both bacteria are gram-negative, capable of producing endotoxins and exotoxins, don't form spores, aren't resistant to boiling, and cause intestinal infections. But they also have differences. Vibrio cholera, unlike Shigella, is a motile bacterium that likes alkaline medium and causes watery diarrhea, rice water stool that can be fatal. Therefore, we will consider the main approaches to the laboratory diagnosis of these diseases. So, let's get started. Shigella, with the ability to produce exotoxin, was isolated from a patient diagnosed with dysentery. What kind of Shigella are we talking about? Shigellosis is caused by 4 types of Shigella, such as Shigella dysenteria, Sonnei, Flexneri, and Boydii. Shigella Newcastle, which is given in the last answer, is a subspecies of Shigella Flexneri, but you should remember that Shiga toxin is present only in first servar of Shigella dysenteria. This toxin is the most dangerous. Shiga toxin, produced by Shigella dysenteria, is a bacterial toxin with N-glycosidase activity. It specifically inhibits protein synthesis in eukaryotic cells by cleaving off the adenine residue of 28S rRNA and thus inactivating the ribosome. Also, as you can see in the photo, it has an enterotoxic, neurotoxic, cytotoxic effect. It consists of 2 domains encoded by chromosomal genes. By the way, Shiga-like toxins also have enterohemorrhagic Escherichia coli, but their formation is coded by the genes of the lysogenic bacteriophage. Other species of Shigella can produce invasins and endotoxins. Endotoxins are released from the bacterium after death and cause non-specific clinical manifestations. By the way, Shigella dysenteria is a mannitol-negative microorganism, doesn't ferment mannitol, and other species are mannitol-positive. So, in this case, the correct answer is A, Shigella dysenteria. The patient was taken to hospital with complaints of headache, fever, frequent bowel movements, and abdominal pain with tenesmus. The doctor diagnosed dysentery and sent the test materials to the laboratory. What method of diagnosis did the bacteriologist have to confirm or deny the clinical diagnosis? If an intestinal infection is suspect, a puke culture must be isolated from the material by a bacteriological method to make a preliminary diagnosis. Please note that the material for research, stool, is inoculated, for example, on endomedium, Shigella salmonella agar. Shigella colonies will be colorless on endos medium because Shigella don't ferment lactose. On Shigella salmonella agar, their colonies will also be colorless, unlike Salmonella. A pure culture is sown on Olkenitsky 3-sugar agar for the purpose of studying biochemical properties. Shigella on it ferment glucose, but don't ferment lactose, so the slanted part remains pink. Thus, a bacteriologist will be able to isolate a pure culture of the pathogen using the bacteriological method. So, in this case, the correct answer is B, bacteriological. At primary sowing of feces on 1% peptone water and 6 hours on the surface of the medium grows, gentle blue film is revealed. Which pathogen is characterized by such culture properties? Vibrio cholera grows well on alkaline elective nutrient media, alkaline midpeptone agar and 1% alkaline peptone water. After 6 or 8 hours, the pathogen forms a delicate blue film. This type of growth is characteristic only of the causative agent of cholera, so the correct answer would be A. Determining the serovariant of Vibrio cholera allows identifying the isolated strain and monitor the epidemic situation. How is it done? An approximate agglutination reaction is placed on a glass with cholera-specific O serum and grown colonies of the pathogen. If the reaction is positive, the other colonies are subjected to an agglutination reaction with typical specific sera of Ogava and Inaba. A positive result of this reaction, together with morphological and culture properties, provides a basis for a positive response. Agglutination of chicken erythrocytes is an express method for determining the causative agent, but not the serovar. By sensitivity to polymyxin, we can distinguish the Altor biovar from the classic biovar. We can determine the biochemical group of the causative agent according to Heiberg's table, and the serovar only using the agglutination reaction. So, the correct answer is B. Agglutination reaction. Vomiting of a patient with suspected cholera was delivered to the bacteriological laboratory. The drug Henhindrop is prepared from pathological material. What method of microscopy will be used to detect the pathogen by its motility? The causative agent of cholera has one polar flagellum, that is, they are mobile. This arrangement of the flagellum is called monotrichial. Henhindrop and wet mount technique are studied using dark field or phase contrast microscopy. In this case, the specimen cannot be treated with dyes and fixed, because this won't allow studying the motility of the microorganism. You can see the techniques of making specimens using these methods in my videos. Other types of microscopy, which are indicated in the answer options, require processing of the specimens and are used for a different purpose. Therefore, the correct answer is D. Phase contrast microscopy.